Hello, everyone. Today we're going to do a lab on limiting reagent. In our lab, we're going to be using two reactants, calcium chloride and sodium carbonate. The first thing that we're going to do is dissolve up both of our reactants to form two solutions. Then we're going to combine those solutions. When those solutions combine, it will result in the formation of a precipitate known as calcium carbonate. This is very similar to what you would think of as chalk, like sidewalk chalk. Now, after we create our precipitate, we're going to need to filter it, dry it, in order to isolate the precipitate from the rest of the solution. To do this, we're going to use a filtering setup. I'll show you what that looks like later on. Let's get our first sample and its mass. Start by getting the mass of an empty 250 milliliter beaker. Okay, so as you can see, the mass of our beaker is 104.28 grams. I'm going to slowly add calcium chloride until I have between 2 and 3 grams of calcium chloride in the beaker. Perfect. So we wound up with a mass of 106.45 grams. We are now going to weigh a second beaker. This one we're going to use for sodium carbonate. Notice that the mass of the second beaker is different than the mass of the first beaker. Every single beaker is made differently, slightly, based on the amount of material. So you're not going to see two beakers that have the exact same mass necessarily. So here is our sodium carbonate. And again, we're going to add about 2 to 3 grams of sodium carbonate. Okay, so in this case, we wind up with a total mass of 104.50 grams. So the next step tells us to dissolve each of our compounds in water. Now, because we're not worried about concentration and we're instead using moles, we don't really care how much water we add, but you should know that the more water you add, the longer it will take to filter. So you're going to try and be conservative when you add water and add just enough to make sure that all of your solid reactant can dissolve. So the first thing that I want you to notice is that as a scientist, it's important to always properly label your beakers so you know exactly what solution is in each. Even though they might look slightly different at first in terms of how the substances dissolve, once they are both completely dissolved, they will look identical to each other. And the only way that we'll know what's in the beaker is by the label that we've put on it. Now, many of you were wondering about the stirring mechanism of the hot plate that we used in class in our last lab. The way this works is that there's a magnet underneath this white plate. What we'll do is we'll drop another magnet in the beaker of our solution. When I turn on the stirring mechanism, the magnet under the plate will turn the bar magnet that I dropped into the solution, stirring our solution. This is what it looks like from above. Speed at which the magnet rotates can be controlled from these knobs. Once our two solutions have completely dissolved and there's no solid present, we can combine them in our third beaker. To keep the magnet from pouring into our beaker, we can use what's called a stir bar retriever to pull the magnet out of our solution. We're now going to transfer the contents of each beaker into our larger beaker, which we have labeled with the chemical reaction that we expect to occur. So we should see the production of calcium carbonate solid. I've transferred all of my calcium chloride in. Now you can see the white solid form as I combine that with my sodium carbonate. This new solution should also stir for a few minutes to ensure complete mixing. So I'll drop my magnet back in and put it back onto the stirring plate. If we take a look at our solution, you'll see a thick precipitate starting to form on the bottom of this beaker. What we're going to do next is set up our filtering apparatus. Okay, so to set up the filtering apparatus, you're going to begin by attaching an iron ring to a ring stand. So this metal piece here is called a ring stand, and this portion here is called an iron ring. So now we're going to filter out the precipitate from our solution. It does not matter which beaker you use. So we're going to label the beaker as waste. We'll put it down here. And then we're going to add our funnel. This is a funnel into our small iron ring. That's going to support our filter paper. And we're going to collect the waste, the sodium chloride aqueous, down here in the beaker, which we'll then discard later. Now, our precipitate is going to be collected in a piece of filter paper. So the very first thing that we want to do, here's our piece of filter paper. We want to get our mass of filter paper. So I've recorded the mass of the filter paper on the filter paper itself, but you should also write it down in your data table. Once you get the piece of filter paper, you need to form it into a specific shape. To do this, fold the filter paper in half once. Like this, and then in half a second time. So it now looks like a quarter of a circle. Then what you want to do is open up the filter paper. So if I hold it like this and I open it up, you want to squeeze it together so you form a cone. So that cone is going to sit in our funnel. And then to help it stick to the side, we're just going to wet it with a little water from the sink. Now, again, we don't care about adding excess water because we're only concerned about the solid precipitate. Okay, so at this point, you should have something that looks like this. Notice how the filter paper is stuck very nicely to the walls of the funnel. You want to make sure that you don't ever poke the filter paper with anything that's sharp. Otherwise, your solid product is going to run through into the bottom. Now, there's another important part to filtering. When you're pouring through the solution, you want to pour it so that it always stays below the top of the filter paper. If the solution that you're trying to filter goes above the level of the filter paper, some of it is going to run over the filter paper and travel down into your beaker below between the funnel and the filter paper itself. And you'll know this happens because your filtrate at the bottom will start to get cloudy. Now, if I back up my camera here and show you at the bottom, if you've done it correctly, your solution at the bottom should be completely colorless. It should look just like water. Now, we know based on our chemical reaction that there's actually salt water mixed in here. So you're going to continue to filter until all of your substance has been passed through the filter paper and collected on top. We've now filtered out almost all of our solid product. Now, we don't want to lose any of it. So to get those last bits of solid product out and into our filter paper, we're going to use a wash bottle, which looks like this. And we're going to spray the water into the beaker. So I'll try and hold it so that you can see this. We're going to spray our water into our beaker and then have the runoff of the beaker go into our filter paper. We're going to hold the beaker in one hand and use a wash bottle to squirt out any of the remaining precipitate into our funnel, through our funnel, and then into our eventual beaker. So now we're just waiting for the last parts to pass through the filter paper. I want you to notice how nice and clear our solution remained down here. That tells us that none of our calcium carbonate precipitate passed into our solution. So once you do this, you can discard of your aqueous sodium chloride in the sink. Once all of your solution runs through the filter paper, you can remove the filter paper from your funnel, and then you want to allow it to dry completely. So to do this, you're going to open up the filter paper. And it will look like this when it's all open. Once it's all done, you could take the mass, final mass of your filter paper and calcium carbonate precipitate.